All right. Hi, everyone. I'm Molly. I'm a second year BISB student in Jonathan Sabat's lab, and it is my great pleasure to introduce Dr. Genevieve Konopka. Dr. Konopka is a professor of neuroscience and the John Hyten Scholar in Autism Research and Townsend Distinguished Chair in Research on Autism Spectrum Disorders at UT Southwestern Medical Center. Dr. Konopka received a dual Bachelor of Science degrees in Brain and Cognitive Science and Biology from MIT and completed her PhD in Neurobio at Harvard University. The Konopka lab focuses on understanding the molecular pathways important for human brain evolution that are also at risk in cognitive disorders such as autism, schizophrenia, and Alzheimer's disease. Her lab uses a combination of human neurons, animal models, and primate comparative genomics to uncover human-specific and disease-relevant patterns of gene expression. Dr. Konopka is particularly interested in the molecular mechanisms underlying language, and part of her research has focused on the FOXP family of transcription factors. The Konopka lab is also part of the International Human Cell Atlas effort to map gene expression in every cell of the human body. This is by no means an exhaustive description of all of Gina's contributions to the field, and I'm so excited to hear more about it from her today. So with that, please join me in welcoming Dr. Genevieve Konopka. Take it away.
Thanks so much for that great introduction, Molly. Thanks for hosting me. Thanks for the students for inviting me. And I'm gonna, I guess, use this laser. I hate to change anything so people on Zoom can see too. Okay, can we see it everywhere? Okay, great. I've had some great meetings. Happy to share some work with you today and happy to take questions at the end. Okay. So the big picture question my lab is interested in addressing is genetic and molecular mechanisms of cognition. So how do we do that? We are asking what features of cognition might be unique to humans, and then whether these features are related to the emergence of what we think of as human-specific disorders, such as autism or schizophrenia. So the work starts from this hypothesis. It's not our hypothesis. It's been in the field for some time that vulnerability to disorders of cognition emerged as a consequence of the evolution of cognition itself. So by studying evolution, we'll get insight into cognitive disorders and vice versa. Now, we're taking a very different approach than some of our colleagues in the field. We're very focused on trying to understand cell types. And as a card-carrying neuroscientist, I feel like I always have to show a picture from Ramon y Cajal. So since the time of Cajal, neuroscientists have been documenting the diversity of cell types, for example, in the neocortex, as well as other brain areas. But I don't think that Ramon y Cajal could have ever imagined really the diversity that we could start to understand when we started to build some of these, and I was not involved in these papers, maybe some of the people in the room were, the brain cell census atlas papers that came out last year in Science. So we've come a long way from cataloging based on morphology to now using transcriptomic methods. And so I just like to, because I know everyone here is coming from different backgrounds, engineering and computer science, just give you some numbers about brain cells. So in the human brain, there are approximately 170 billion cells. So to think about what that number means, that's almost on par, that's roughly on par with the number of stars in the Milky Way. So if we're trying to understand the diversity of cell types in the human brain, this is a large space, a large number of entities to deal with. And as I already mentioned, even though we started over 100 years ago documenting diversity of cell types based on morphology from the times of Cajal, we still don't know all of the cell types and how they connect and how they function. And so using more modern methods such as rainbow mice, we're able to understand how cells were connected to one another. But the level that my lab has been looking at is trying to understand this from the genetic and genomic level. So now here we have another large number to deal with, right? So what's remarkable to me is that every cell in your body has the same DNA, yet how that DNA is harnessed to lead to changes in gene expression and ultimately cell identity and function is quite diverse. And so we have roughly 3 billion letters of DNA that code for over 20,000 genes in any given cell in your brain. Maybe you have 8,000 of those genes expressed at any given time during development. So this becomes a really challenging computational problem that all of you can continue to work on with us as we move forward. So the question is, how can we understand, my lab is asking, brain function, evolution, and disease at this cell type level? So today I'll share with you 2 vignettes of recent work from the lab. The first is using a single cell approach to compare human versus non-human primate neocortex. And the second is our work where we're integrating genomics within human brain measures and behavior. So we'll start with this first one. So this comes from this question of looking at human and non-human primate brains, which the field has been doing for some time. This is a human brain. This is a chimpanzee brain, our closest existing genetic relatives. And if you just look at, other than the obvious difference in size, this is to scale, they're very similar at a gross level. So how can we understand the dramatic differences in phenotypes that humans and non-human primates have, right? We're studying them, they're not studying us. We're sending satellites up to space. We're building art and music. So there must be some other feature embedded in us that's driving these phenotypic changes. And we think that it could be at the level of molecular mechanisms. And a lot of this work stems from a very, I think, important observation that was made now almost 50 years ago by Mary Claire King and A.C. Wilson. They were comparing proteins of humans and non-human primates, as well as other species. And remember, we didn't have genomes yet. And they made this very important observation saying their macromolecules are so alike that regulatory mutations may account for their biological differences. And then we knew, moving forward decades later, upon the sequencing of the human and the chimpanzee and other genomes, that indeed, at the level of coding regions, we are very similar to chimpanzees. So there must be something else that's driving differences in molecular phenotypes. And so gene regulation is where we and others have been looking. And so for a number of years, we've been comparing 3 species, so human and chimpanzee, and then we use rhesus macaque as our outgroup, which the common ancestor diverged over 35 million years ago. So if you compare these 3 species, you're mostly going to observe changes that have occurred on the macaque lineage due to evolutionary distance. But what we're trying to look for are things that are similar between chimps and rhesus macaque, and those that are then different specifically on humans, so-called changes on the human lineage. And so many years ago, we started doing this with microarrays, and then we started doing bulk RNA-seq. And before we had true single cell genomics, we could do something where we could sort out cell types. Because again, thinking back to the era of Cajal and this diversity, we thought surely there must be differences if we could break apart the tissue into different cell types among the species. And so this was a project that was published now, it's 5 years ago. I had 3 very talented postdocs that worked on this, and we obtained a tissue from dorsolateral prefrontal cortex, Brodmann area 46. This is an area of the brain that's thought to have been enlarged in human evolution. It's also frequently at risk in many cognitive disorders like schizophrenia. And so we had postmortem tissue from humans, so unaffected individuals with schizophrenia, because we're interested in disease risk, and then tissue from chimpanzees and rhesus macaque. And then we could use an isolation method for nuclei. These are postmortem frozen tissue so that you can't get intact cells once it's frozen. And then we could isolate broadly neurons using an antibody to nu-N, and then cells on the oligodendrocyte lineage using an antibody to olig-2. We could sort these out, and then we could do RNA sequencing and from dozens of individuals. And if we just look at the kind of first result, principal component analysis, we could recapitulate phylogenetic differences for both the neuronal data and the oligodendrocyte data. So that was good. So it seemed like things were working. But what we're really interested in are things that are different, things that were changing specifically in humans, and we needed to take into account evolutionary distances. Because again, remember, we'd get mostly changes in macaque if we didn't account for that. So when we did that and took into account millions of years difference and differential expression, we could identify the genes changing specifically on the human lineage in the neurons, as well as in the oligodendrocytes. And what was striking to us was, 1, that we found so many changes in oligodendrocytes, and 2, it even seemed to be more robust than what we found in the neurons. This was surprising because all of the prior work from my own lab and from others in the field had primarily observed changes in neurons when comparing humans to non-human primates. So I took these data with a large dose of skepticism and decided that we needed to investigate this further. So, oh, someone's drawing on the screen. Cool. So the first thing, not me, the first thing we did was we compared our differentially expressed genes to the genes that had been previously identified in the other studies. And as I mentioned, previously we'd seen changes in the human lineage in neurons. And indeed, we could significantly overlap these new sorted data with a previous literature. And our oligodendrocyte human-specific genes did not overlap with anything from the previous studies. Again, it was true. We really hadn't found changes in oligodendrocytes before. But this was still not satisfactory, that, even if we hadn't found it before, maybe the results were there. And so right around this time, a few studies had been applying single-cell genomics to brain tissue, primarily from the Allen Brain Institute. These were kind of early days. And so what we could do is we could take those kind of broad cell type classifications that they had identified at the single cell level and deconvolute our bulk data and the previous data to infer what was going on. And so we deconvoluted these 4 previously published data sets from bulk tissue. And what we found was that the signal was primarily being driven from all the species from excitatory neurons. So we were essentially underpowered to detect anything coming from oligodendrocytes in these previous studies, which is why we had not found the differentially expressed genes. We now know from many years of us and others doing single-cell RNA sequencing from brain tissue that neurons express many more genes and at higher levels. So if you just take a chunk of tissue from the brain and don't do a single-cell profile, from the cortex at least, I should say, you're primarily going to get a result driven from neurons. We applied the same deconvolution approach to our fan-sorted data, and nicely, we got, in our new end data, signal driven not only by excitatory neurons, but also inhibitory neurons, and the OLIG2 data was driven by oligodendrocytes. So our sorting had worked, and the differentially expressed genes that we had identified, I believed, were real, and we just simply had not been powered previously to detect them. So this was quite a new and exciting result for us. We then wanted to try and prioritize, well, among all these genes that are differentially expressed, specifically in the human brain, what might be some of them that we might want to follow up functionally in the lab or the field among all of the hundreds of genes that were changing. And so to do this, we applied weighted gene co-expression network analysis. This is an approach to look at how groups of genes are co-expressed across all of your samples. It's not typically used these days in single cell approach for a number of reasons we can talk about afterwards. But for these sorted data and for bulk tissue, it's a really great way of prioritizing key hub genes. And so we applied this to our data, and we could identify 2 human-specific neuronal modules. So a module is a group of co-expressed genes, and 2 human-specific oligodendrocyte module, human-specific oligodendrocyte modules. Okay, so that's great. So what does that mean? So we can visualize the genes within these modules, and this is just one example of a human-specific neuronal module, and each one of these red circles indicates a gene that was differentially expressed. You can see some gray genes. These are genes that weren't technically differentially expressed, but are co-expressed with the DEGs. So this is a way to add more genes in a pathway, if you will, or to understand more the overall pattern of gene expression. Then we could run one of my not so favorite things to do, gene ontology to the data. And not surprisingly, from neuronal sorted nuclei, you get genes that are involved in vesicle-mediated transport, synapse, things like that. Okay, so that's really cool. But what I was really interested in was the oligodendrocyte, because this was really a novel finding. And so this is one of the human-specific oligodendrocyte modules. Again, all the red genes are the differentially expressed genes. And if you run the gene ontology, in this case, now you start to see mRNA processing and splicing. And to my knowledge, there's very little work done on RNA processing within oligodendrocytes. There's a lot of work being done in neurons. So to me, an unanswered question would be, maybe there's something unique about human oligodendrocytes such that RNAs are trafficked or processed in a way that we previously haven't appreciated. And so I've been showing this slide now for a few years, and I keep hoping that someone's going to follow up functionally on one or more of these genes in a human iPS-derived oligodendrocytes. Maybe my lab finally will do that this year. But I think it's very, these are promising leads to follow up from an evolutionary perspective and for this understudied cell type within the mammalian cortex. Okay, so those were the key findings from this previous study with respect to evolution. But remember, I told you that we're very interested in how disease and evolution may intersect. So we want to get back to this idea of this potential trade-off of brain evolution putting us at risk for cognitive disorder. And so the first thing we did, which was a bit simpler, I suppose, was that just to take the data and look for enrichment from brain-associated GWAS, genome-wide association study data set. So we could take at the time all of the available brain-related GWAS and then as a control non-brain GWAS. And what was a bit disappointing to us, considering the number of DEGs that we had in the neurons, in the human-specific neuronal modules, we didn't find any enrichment for DNA GWAS signals. However, in contrast, when we compared the human-specific oligodendrocyte modules, we found a significant enrichment of brain GWAS. The negative control was negative among a number of cognitive disorders, including like bipolar and ADHD and schizophrenia. So this was really great. And I think it's really a high bar to overcome when you're comparing kind of, a DNA signal to this cell type specific differential expression, co-expression signal. So you're not exactly comparing apples to apples. So the fact that we even got a significant enrichment to me was, I think, really promising. But if you were paying attention to about 5 slides prior, I mentioned that we had also done the same exact sorting experiment using tissue from individuals with schizophrenia. So we'd done the same exact process, isolated the neurons and the oligodendrocytes in controls in schizophrenia, identified differentially expressed genes at a cell type level. So we could compare those DEGs to the human-specific DEGs. And what we found was the neuronal genes that were differentially expressed in schizophrenia did not overlap with the evolutionary signals. But again, we found this really amazing result in the oligodendrocytes. So the genes that were significantly downregulated in oligodendrocytes in the tissue from individuals with schizophrenia were the genes that were significantly upregulated in the human brain compared to non-human primate brain, and vice versa. So this really supported this idea of this evolutionary trade-off, at least at the gene expression level, at the cell type specific gene expression level for oligodendrocytes of evolution and schizophrenia pathophysiology. Okay, so around this time, so we were super excited. I never thought that I would be as excited about oligodendrocytes as I was at that time. And around the time of our paper being published, a study from Philip Pajdovich's group had been starting to now apply single, drop-seq 10x genomics type single cell RNA sequencing to evolutionary data sets. In this case, they had human, chimp, and macaque, but also bonobo. And they derived this measure of what they called a human specificity index for differential expression. And what was really, and this paper had multiple brain regions, but I'm just showing the data from the anterior cingulate cortex. This is an area in the brain that's important for theory of mind and other cognitive processes. And what they found was that the greatest amount of human specificity at their kind of major cell type level, right? This is early days of single cell, was driven by signals from non-neurons, in particular oligodendrocytes. So this fit in really well with what we had found from our dorsolateral prefrontal cortex. But as we started to dig into this and as the technology became improved, we decided that we wanted to understand if there were kind of subtypes of oligodendrocytes or subtypes of neurons that might be have evolved specifically in the human brain. And so to do this, we carried out 10x single cell RNA sequencing and also attack sequencing, not true multi-omi yet, that wasn't available, but independent. And this was the work of a very talented graduate student, Emory, who graduated last year and is now a postdoc at Chris Walsh's lab at Harvard. And for Emory's project, we compared, again, tissue from human, chimpanzee, and rhesus macaque. In this case, we were using the posterior cingulate cortex. Why the posterior cingulate cortex? Well, it has clear anatomical homology among the species. It's a region that's critical for higher order cognition. It's been known to be disrupted in individuals with schizophrenia based on imaging studies and other metrics. And just as a reminder, again, we're using postmortem tissue samples. And so we're have to do the nuclei isolation. Okay, so Emory was already primed from our previous work to be looking at oligodendrocytes. And so I just give away the take home here that he found a significant change in oligodendrocytes, specifically in the humans. So here we have our posterior cingulate cortex. Now with more modern single cell resolution, we could identify all sorts of subtypes of cells along the oligodendrocyte lineage. And one of the first things that Emery noticed was that there appeared to be way more of these immature oligodendrocytes, so committed oligodendrocyte precursors or oligodendrocyte precursor cells in the human data set. But he wanted to be robust about it and statistically assess this both from the RNA-seq as well as the ATAC-seq data. And what he found was that there was a significant increase indeed of the immature, the OPC relevant oligodendrocytes in human, and a concurrent significant decrease in the mature oligodendrocytes. So we thought this was super exciting. This change in proportion had never previously been reported before, which also made me skeptical whenever you find something new. And so one of the issues that we had to, I think, first address and get out of the way is the fact that when you're taking these postmortem tissues and isolating nuclei from them, you're basically taking the cells out of their spatial context. So it was possible, even though we had matched everything to the best of our ability, we're matching by humanized age, by sex, by, batching all the processing. What if by chance we took more of, say, the white matter below the cortex of one or more of the species versus the other? What if we had just done a dissection that wasn't exactly equivalent? We wouldn't know that because we were isolating them. So could we look in intact tissue? So to do this, we applied single molecule FISH, RNA scope, and we got tissue from independent human and chimpanzee samples. And we looked throughout the entire neocortical layers. We didn't see any, it was robust to cortical layer. And we were able to recapitulate the single nuclear RNA-seq finding with this increase in the immature oligodendrocytes, as indicated by PDGFRA, and a decrease in the mature oligodendrocytes as indicated by signal for MOG. So using this intact tissue pieces, we could recapitulate this change in proportional signals specifically in humans. So I was really excited by that. But then the ever skeptical person in me was like, well, what if this is just something specific to the posterior cingulate? No one really looks in the posterior cingulate cortex genomically. There's something a little bit different about the lamination in the posterior cingulate cortex in humans. How robust would this finding be to other areas of the brain? And so to do this, we decided to, first of all, reanalyze the Kaitovich paper where they had looked in the anterior cingulate cortex. And indeed, we could recapitulate our finding in their anterior cingulate cortex data. Now, you may say, well, why didn't they observe this change in proportion? Well, one, they weren't looking for it, but two, also along the way, Emery had determined that the field was potentially missing a very important technical artifact to all of this tissue processing. Namely, if you take, in particular, a human brain where the neurons are quite large. I don't know if you remember from the Cajal picture I showed you, lots of processes, and you're dissociating this to obtain your nuclei. There are RNAs, as we know, getting trafficked, right, throughout the axon and dendrites. And as you're breaking those tissues up, especially the neurons will release all of those RNAs into your solution. And those free-floating RNAs can get encapsulated during your droplet formation into what is called so-called ambient RNA. And so, in fact, at one point, Emery thought he had discovered a new cell type, which was a combination of kind of an oligodendrocyte and a neuronal signature, but it was just neuronal ambient RNA contamination. So he got a nice neuron paper out of that too. How to clean your data, observe it, and clean it. So we cleaned up, yeah, this is good. It's a win-win. Kaitovich's paper, we could recapitulate our finding, and then we got, again, independent tissue, independent individuals, anterior cingulate, did the RNA scope, and could recapitulate this finding of this change in the ratio of immature to mature oligodendrocytes. So this was great. So we had posterior cingulate cortex, anterior cingulate cortex. Again, it's like, well, what if this is just something about singular cortex? Will anyone care? What about other parts of the cortex? So we had available to us our fan sorted data that I started the talk with, where we couldn't exactly do this at the same level, but we could look at the ratio of OPC to MOL from deconvoluted data. We could recapitulate that in the dorsolateral prefrontal cortex. The Allen Institute and their colleagues at this time had already started to publish some more single cell data here from the primary motor cortex. In this case, they didn't have chimpanzee, but they had rhesus macaque and marmoset. Again, we could see this change in proportion. So everywhere we looked in the cortex, we could recapitulate this change in proportion. So I was like, this is great. It's robust. Even if we use a marmoset instead of macaque, we see it. What about outside of the cortex? Is this anywhere within the human brain? So at the time, what was available to us were data sets from the caudate nucleus and the dentate gyrus. Again, here we have some chimpanzee or great ape and some rhesus macaque. And in these brain regions, we did not see a difference in ratio. So of the areas which we've investigated, we've seen this change in OPC ratio, specifically in the cortex. Doesn't mean it's not occurring in other parts of the brain. We haven't looked everywhere. And as we and others are generating additional data sets in human and non-human primate, we'll be able to understand the extent of this, this proportional change. So this is really exciting. It had never been observed before, but still the big question is, why? Why would this have evolved? What is it doing? And I don't have an answer to this, but I have kind of, just as a reminder for those in the audience that may not think about oligodendrocytes, and certainly it wasn't on my radar to a few years ago, these are not only used, certainly they're important for myelinating the axons, right, that are required for sending signals over long distances, but there's a whole burgeoning literature out there in how particularly immature oligodendrocytes, OPCs, are interacting with neurons in ways such as synaptic pruning, as well as sending and receiving signaling molecules between the oligodendrocyte lineage and neurons. So they're not only there to provide a source of myelination. There's also, it's much more complicated than that. We also understand now that astrocytes and microglia are playing a much more important role than we previously appreciated. So what is our hypothesis? Our working hypothesis is that having this increased ratio, this increased pool of OPCs sitting around, is allowing for an enhanced potential for experience-dependent generation of mature oligodendrocytes. So the pool sitting around, a signal comes in, you learn something new, your OPCs can respond in a way to support the neurons, not necessarily through myelination, but in however way they're interacting with the neurons. And I think this may underlie our continued ability to learn new things as we get older. At least I hope that's what that means, that we can still learn new things. And that it may explain some of the differences in aging that we see and abilities that we see in aged humans compared to non-human primates, as well as the enhanced susceptibility to neurodegenerative and other diseases in the aged human that we don't see in a non-human primate spontaneously. So how are we going to test this? Well, we're going to do things in culture with iPS-derived oligodendrocytes and neurons from a number of species. We're also really interested in trying to understand differences in oligodendrocytes and white matter versus gray matter. Everything I showed here to you today was in gray matter parts of the brain, right, where there's a lot of neurons. But in white matter, is primarily oligodendrocytes. So and the developmental trajectory of oligodendrocytes in white versus gray matter is very different. So now these were all adult tissues. We're looking over development, white versus gray matter across these species to try and understand the development, and also in disease and autism, we have tissue to do that. So a lot of different ways, a lot of different angles to try and understand why this has evolved and what is it doing functionally. Okay, now, we also had neuronal data in this data set. I don't want you to think we only focused on oligodendrocytes. Here are just some the UMAPs from the excitatory neurons. We had more excitatory neurons than inhibitory neurons for whatever reason in these data sets. And I mentioned we also had the single cell attack sequencing data. So not only could we be looking at changes in expression, but we could also be looking at changes in chromatin accessibility across the species. And what we really wanted to get at was what could be going back to King and Wilson, what are the regulatory mechanisms for driving changes in gene expression? Could we identify factors that might be driving cell type specific expression patterns in the human brain? So we could do this by mining the attack sequencing data. Okay, so here's just, this is just a subset of everything we found. I can refer you to the paper, but this is from the excitatory neurons looking for motif enrichment from, let's see, this is lower layer to upper layer neurons. And what Emery observed was two key things. One was that the upper layer excitatory neurons, when we're looking at the differentially accessible regions, so these would be human-specific, open, cis-regulatory regions, looking for motif enrichment. So FOS-Jun motifs were enriched in the upper layer excitatory neurons. In contrast, FOXP factors, notable FOXP2, was enriched in the lower layer excitatory neurons. Now, this was really crazy because my lab also works on FOXP1 and FOXP2. FOXP2 is a gene that's been implicated in human brain evolution with respect to speech and language development. And so now we have evidence for regions of the human genome that are uniquely accessible, or I should say uniquely accessible and have motifs for FOXP2. So those would be areas of further functional study. But Emery, since he didn't have the history of being excited about FOXP2, he was more excited about the FOS-Jun motifs because these are motifs that are involved in kind of immediate early gene activation. So these are motifs that can respond to signals in a real-time manner. So this gets us a little bit closer to thinking about evolutionary changes that are important for activity-dependent gene expression. But he was also well aware that FOS-Jun themselves are very susceptible to changes in expression. And so if they were just changing due to whatever postmortem interval or something like that, or how we obtain the tissue, maybe that it could be an artifact. We're always worried about artifacts. And so the first thing he did was just to look at the expression of FOS and Jun themselves in all of the cell types across all of the species. So this dot plot indicates the amount essentially of expression. And hopefully you can appreciate that all the dots look similar across all of the cell types. So there was no change in the expression of these factors themselves. But what we couldn't account for was that the postmortem interval is quite different between the human samples, which are obtained from people after they pass away. Some time elapses until the family agrees to have tissue donation, compared to the non-human primate samples, because those animals were in primate facilities that were monitored around the clock, right? So they have very low PMIs. So how could we address this? So the best way we could do it was to obtain human samples that had a post-mortem interval of 0. I don't mean rapid autopsy, I mean individuals that are actually still alive. So they have a surgical intervention and their tissues removed, and we can carry out the same sort of sequencing. And we already had one of these data sets in hand from a previous study, which is actually the second part of my talk. And so what Emery could do was to mine that data, looking for the enrichment of the motifs, substituting this human data for the postmortem data, and essentially found the same thing: enrichment for FOS-Jun motifs in the upper layer excitatory DARs. So this was great. So this result likely had nothing to do with postmortem interval. We believe it was robust. And then just to try and add some additional layer of understanding to regulation, Emery looked for evidence of human accelerated regions within the human-specific open CREs. He also took into account available ancient human genomes, so Neanderthal and Denisovan genomes, so he could assign within these differentially open regions, were there variants that were specific to modern humans. And so here's just an example of a FOS-Jun motif that is novel, that falls within a HAR within one of these human-specific open regions. And where is it, right? What might it be important for this kind of responsiveness to activity? Well, it's upstream of this gene, MTHFD2L. So what does that gene do? It's involved in the folate cycle, and there's a whole literature in comparative primatology, if you will, and neuroanatomy and anthropology that human brains may have evolved a higher, for various reasons, higher metabolic need. And so we think this is a great example of a specific variant that's evolved within this human-specific accessible region in a cell type specific manner in the brain that might be important for neuronal activity or metabolism. So this is just one example. There are many from within the paper. So this is really exciting. But again, all of these data are essentially from postmortem tissues. And what, as I've said to some of the faculty, my long-term goal is to really understand how gene expression in the human brain is at play during functional tasks in a living system, not as a snapshot after some time post-death. So how can we do that? Well, this work stems from a collaboration I've had for a number of years with a neurosurgeon at UT Southwestern, Brad Lega, who has one of the busiest epilepsy surgeries and surgical practices in northern Texas. So these are patients with treatment, medication-resistant epilepsy, and so they come to see him to map the site of their epilepsy, and then in some cases, tissue gets removed. And in the meantime, he's also carrying out a number of tests on them, one of which is measuring oscillatory behavior during specific memory tasks. So the question then was, could we use some of our genomic methods to identify human brain activity genes related to a memory task? And so how do we do this? So these are patients that are implanted with these electrodes. Here, these green dots are just showing you everywhere the electrodes are placed. And the patients are shown words on a screen while they're measuring their oscillatory activity. They then are given a math distractor task, and then they're asked to recall the words. And so all the time they're measuring oscillations, and they can calculate this subsequent memory effect based on accurate recall and what the oscillatory activity looks like. And so the first thing we did, this is a paper published a number of years ago, again, 2 great postdocs, worked on it with Brad, was to correlate this intracranial EEG activity with measures of gene expression. Except in this case, we were using gene expression data from postmortem tissues. We were powered in this case because we had data from many cortical areas, postmortem and intracranial EEG, so we could look at the correlation of the gene expression and the oscillatory signals across the brain regions. And what we were able to identify were genes that correlated with accurate memory recall oscillatory signatures, and most of these fell within the range of beta oscillations. So this was exciting. And we also did it with resting state fMRI data. There are a couple of other groups that did that. But the major caveat was that we're comparing data from independent populations, right? The gene expression was from postmortem tissue. The intracranial EEG was from patients with epilepsy. And so I'm always thinking of how I can push the envelope further. How can we do this from the same population? And so as a consequence of this, this particular form of epilepsy, what Brad often has to do is remove the anterior temporal lobe, often the amygdala and the hippocampus, to get within the brain to the site of where the actual epilepsy is. So this would be considered quote unquote normal tissue that's kind of physically in the way to be removed. I recognize that these patients have had epilepsy. They've had epilepsy for most of their life. They've been on medications. We take all of that information into our covariate modeling, but it's still not the tissue where the seizure is happening. And we would exclude any tissue that had sclerosis or heterotopia or any sort of abnormal pathology. So a lot of many patients were put into this study and we couldn't use their tissue. And it took us well over 5 years to accumulate all of the data that we actually needed for this study. So this was the first within subject study of gene expression and intracranial EEG. And so these patients were recruited that had medication-resistant epilepsy. They have the electrodes implanted. They undergo the EEG and memory testing, and they also undergo a number of brain imaging, MRI and fMRI. And in some cases, the tissue is resected, and in some cases, the tissue can be used for genomics. And then we can correlate all of these metrics. Okay, and again, we're using the same memory task with words on a screen, this verbal free recall. But in this case, instead of mapping across the entire brain, we're not getting tissue from the entire brain. These people aren't volunteering to give us all of their brain tissue. We're getting only tissue here from the temporal pole. So we're only able to use the electrode data from the temporal pole and the gene expression from the temporal pole. But we're comparing within subjects. So the same individuals are contributing both types of data. So here's the overall study design. So we did the tissue resection. So over 5 years, we were able to collect data from 16 individuals that could give us good gene expression and good intracranial EEG SME mapping. We also had 11 participants where we got good gene expression data, but not good mapping data. And then as some additional controls from frozen brain tissue, We had just postmortem tissue from healthy individuals and individuals with epilepsy, because we weren't interested in trying to map how oscillations were related to epilepsy. We're trying to understand how this is mapped to accurate memory recall. This is like a basic normal condition we're trying to look at. Okay, so then we could correlate the gene expression in the SMEs, and we could find the genes that, and use all of these control data as permutation controls, and then narrow down to a list of genes that are associated with accurate memory recall. And then we also carried out this co-expression network analysis, and we also did additional single cell data analysis to the data. Okay, so what did we find? Well, here's just a heat map of all the genes that were correlated with different wavelengths of accurate memory recall using a multivariate analysis. And then the reviewers asked us to kind of be more robust. So we carried out Spearman's rank correlation and narrowed this list down even further. And in the end, we were left with a roughly 300 genes that correlated with accurate memory recall at these different wavelengths, with the majority of the genes correlating with low frequency delta oscillations. So I showed you a few slides back. We're mapping across the entire neocortex. We found beta correlated genes, but when we're focusing just on this one region, we found just delta, which was good. I mean, we were hoping we wouldn't keep finding consistently the same things, because we were doing a very different experiment. Okay, and then we also had some very other important parameters, again, to control for the fact, well, maybe this is just the noise from the data. So we had the oscillations while the patients were performing the math distractor task. We could correlate genes. We had just their kind of quantitative memory performance overall. And from the imaging data, we had cortical thickness. And hopefully what you can appreciate, with the exception of one gene, all of the genes correlated to human memory performance related to oscillations were unique. So this was great. Okay, then again, we're trying to identify and prioritize specific genes. This is a very busy slide. I'm going to just walk you through what you want to look at. We did the weighted gene co-expression network analysis. We wanted to see where those genes fell with respect to other diseases or other studies. And this module of co-expressed genes, WM12, was of interest because here were the genes that, again, were these kind of most robust delta memory relevant oscillatory genes. And they also significantly overlapped with our previous study across subjects. So it seemed like these are genes that were coming up kind of again and again a little bit with respect to human memory. And if we visualize the top 10 highly connected genes in WM12, here's a list of them. These were of interest because they were significantly correlated. SHANK2 and SMAD3 run the gene ontology. You see enrichment for voltage-gated ion channel activity. Great. So, we're using brain tissue and we get neuronal genes. Not surprising, right? Especially because I told you at the beginning, like you're normally going to get neuronal gene transcripts popping up because they express more genes. Okay, but what else could we do with these data? Well, we also wanted to see specifically how it overlapped with disease. And so here again is the same group of co-expressed genes. And these were all our favorite brain GWAS. And we found significant enrichment for disorders like ADHD, bipolar, again, schizophrenia. What was very important was here, this EP 2018. These are genes associated with epilepsy. So again, remember, we're not trying to pull out genes associated with epilepsy. We're trying to pull out genes associated with normal memory performance. So indeed, this module was not enriched for epilepsy relevant genetic signals, which is great. And those patients honestly should have been excluded from the study anyway, if they had one of those variants. So this was really nice. So there was disease relevance here. Also, because many of these diseases have memory phenotypes, thinking about, for example, here, autism, although it's not quite at the level of significance, intellectual disability is frequently comorbid with autism. Okay, so we'd found some genes, we did the gene ontology, but again, I've been really focused on trying to identify contributions at the cell type level. So we carried out independent processing of a subset of these patients doing single cell RNA sequencing. This is just a UMAP. From the single cell RNA sequencing. And then we could take that cell type specific information and integrate it with the co-expression network analysis that we had done with the memory relevant genes. And again, here's our WM12, and the cell type that these genes were most significantly enriched for was a specific type of excitatory lower layer neuron, RORB SMA3E positive. I didn't come up with these labels. We can thank our colleagues at the Allen Brain Institute for the nomenclature. So now we have a cell type that we could be thinking about potentially down the road targeting for, neuromodulation for memory relevant processing. So this was really exciting. But we didn't want to stop there. We were also doing single cell attack sequencing. We wanted to know what could be the key factors we could manipulate in a cell type specific manner to drive memory processes. So we did that. Here's just the UMAP of the attack sequencing. And now looking within WM12, we could identify all the significantly enriched motifs within the differentially open regions. And one thing that stood out to us was an enrichment for the motif for SMAD3. So another thing about this was when we looked where SMAD3 motifs were enriched, at least one of them was upstream of SHANK2. And I showed you that WM12, two of the top co-expressed genes were themselves SMAD3 and SHANK2. So all of this was falling into place as these genes being co-regulators, or at least SMAD3, a major regulator of all of the gene co-expression in this memory relevant module. So SMAD3 is a gene that we're actively going after to try and understand its role in memory relevant human brain activity and just gene expression in human neurons in general. So what we were able to do in this study, I think, is really quite remarkable. We were able to take living humans, have them do a behavioral task like a verbal free recall task, correlate this behavior with intracranial EEG measures to identify genes that may underlie this behavior, and then using single cell RNA-seq identify the cell types that might be the most important ones, and then with the single cell attack-seq, the transcription factors that might be co-regulating all of this gene expression. So where are we moving forward with SMAD3? Well, now I have my team of, we call them the human brain team. Haley and Anna are graduate students and Mantra is a postdoc, working with Brad to really try and use the tissue that he's getting from the epilepsy patients to address a number of questions. One is just a general question of how do genes change in the human brain with stimulation. I think this is going to be important to understand mechanisms of things like deep brain stimulation. So you can see immediate early genes go up. But also Haley and team have identified and developed a way of taking this tissue and keeping it in culture. So making organotypic slice cultures that are alive for up to several weeks. So we can do things like chronic stimulation, plus or minus, multi-electrode array, but also for the purposes of what I just showed you, potentially manipulating genes of interest like SMAD3 in these slices and trying to understand at a cell type specific level, what are the gene expression changes downstream of SMAD3 in a human cortex. So this is where we're going over the next 5 years with this project, and we're super excited about it. Okay, so future directions. From the first half, we really want to understand the function of this increased OPC ratio in the human brains. We want to understand the dynamics of activity regulated elements between species, and then overdevelopment and between species. That's where we're going. Okay, so I think I'd thank the key players along the way, but obviously I have other players in the lab that contribute. So I'd like to thank the rest of my lab, my wonderful collaborators, funding agencies. Thank you for your time. Happy to take questions. Colleen? Oh, thank you.
Have you been generating? I'm sure you've done it. Did you guys set the biopsy? Or how many guys in you? What was the balance?
That's a great question. So in his study, we were balanced. We had 2 males, 2 females, and I don't think that's sufficient to like draw enough of kind of an interpretation. But we take sex into it's like one of the covariates we list, but we didn't try and disentangle. But now moving forward in the projects that we're getting funded to do, we have a much larger sample size. Again, we're trying to match male and female numbers. So I think moving forward, we'll be able to answer that. But yeah, it's a great question. Thanks. Yeah, John.
So the oligodendrocyte expression that you're seeing between humans and non-human primates, that you're seeing in schizophrenia, is really cool because it looks like you're seeing a cellular signal or something that we also have fMRI, evidence of in fMRI. Right?
Mm-hmm.
So there's white matter increases in larger brains relative to small brains, macaque versus human. There's schizophrenia, very clearly reduced. There's increased white matter and connectivity in certain parts of the brain, but reduced overall. So to what extent can you correlate the cellular signal with that signal? And then we have genes, right?
Yeah.
How are we gonna? Can we clearly show the genes that can be phenotyped in humans
Mm-hmm.
with real genetics?
Mm-hmm.
How do we actually show that that's really affecting oligodendrocytes and really affecting that way?
Right, because the chicken or an egg thing, what we're observing in the adult postmortem tissue versus like genetic risk, which is why the GWAS integration was nice, but then the gene expression was another layer. But these data can't get at that question of the fMRI signals and white matter because we were stuck in the gray matter. But moving forward, that's exactly what we're planning to do. We're processing white matter tracks from throughout the brain, like 6 different white matter regions and functionally associated gray matter regions in humans and non-human primates. And then we're doing a similar thing in autism from postmortem tissue. But I haven't yet obtained any funding to do it in schizophrenia, but I'd love to. So that's another grant I need to write. But yeah, I think also looking for the specific, strong genetic hits within the data, just it makes sense. And that was something we just, other than running the GWAS and we haven't done that. But yeah, great suggestion. Thanks. Yes. Yeah, that's a great question. That's something that we're also very interested in. So we tried to match, again, 444, the ages as best we could. So we use life history traits in the non-human primates. So it's like age of first incisor, like age of puberty, things like that to kind of match developmentally stage the age of the non-human primates to humans. But I think that's it's what we can do. But now moving forward, what we'd like to do is like methylation and use like the Steve Horvath methylation clock within our samples to more accurately match the ages. But then we also want to do this over development, right? Because I think, as I alluded to, the developmental trajectory, specifically of oligodendrocytes in gray matter versus white matter, at least in humans, is very different. So we don't know how that would compare, say, in a chimpanzee or a rhesus macaque. And then we have to match those over all of these different time periods. And then in aging too. So we're funded for development up to middle age, and then we need to figure out how to get funded to look in aging as well. But yeah, very important questions. Thank you. Yeah. Say that again. As it applies to single cell. Yeah. Right. Because I think, we know when we were applying it back in the day from bulk tissue, basically what we were getting at was cell type information. It was almost like doing a T convolution without realizing we were doing deconvolution, right? So if you're already able to identify the cell types, what will the co-expression get you once you beyond giving you cell type information? So where it's been useful, for example, is work from Aparna Badori and Arnold Krigstein have applied WGCNA, for example, to developmental data sets. It's kind of like using it as a sort of like pseudo time analysis of the data, getting another metric of co-expression in the because cell types and cell states are dynamic, right? So there's additional layers. I think it's more cell state, right, that you could get at from the developmental pattern. So, but yeah, Emery and I have argued back and forth about this. And, and the other issue is the single cell data, as you probably know, has a lot of zeros, right? And so WGCNA does not do well with zeros. And so there are the ways to get around that. You can make a meta cell, or you can do some other mathematical massaging of the data, or you could do imputation, which that's like a bad word in my lab. We're not allowed to say imputation. I'm sorry. So yeah, so I think that's why we've kind of gotten away from it, but I'm open to other ways of analyzing the data. I was trying to see if there's something in the chat. Any other questions? Yes. Oh, I see, kind of like biotyping the patients in a way, or coming with cohorts. Yeah, because we were really focused on kind of, honestly, we were with 16 patients, 16, we're really underpowered, right? I mean, we needed 120 to be able to do that. We did the calculation, so we had to do within subjects, sorry. Reviewers asked us that. Anything else? All right, thank you.